Chapter 1. Chapter 1, we're going to be looking at kind of a variety of things. Stuff we'll need kind of foundational for microbiology. Things we're going to need some basics of that. Some diseases, emerging infectious diseases, some things, the basis of it. We're going to look at some history of microbiology as well. So a little bit of everything you're going to learn from here. So one of the things I usually try to treat is your chapter objectives for each regular chapter, except for the diseases. There'll be chapter objectives. Kind of look at these as what I'm required to teach and what I need to test on. So kind of look at it as a checklist of things you need to make sure you do know about. These are all things that I will test on for sure. Okay, these are things I'm going to be pulling out for my test. Okay, so make sure you are paying attention to those. So as we go on, we're going to start off, because this is microbiology, so we're talking about micro, we're talking about small things, we're talking about things that are too small to see with your eyes, your naked eye, your unaided eye. This is small stuff. We're looking at very small things. Germ is just something, is a cell that rapidly grows. We refer to as a germ. Okay. Some germs are pathogenic. They cause disease. Not all germs are going to cause you to have disease. Not all can affect you. Some you are cause disease in some organisms, some in other organisms, but not in you. There's loads of functions that germs do. They can be decomposers. They can decompose organic waste. So we have that. When we die, our bacteria are going to take our bodies. They can be producers in the ecosystem. They can do photosynthesis and produce sugars. They can produce chemicals in industrial things like ethanol and acetone used in industrial processes. We use them for making fermented foods like making yogurts and cheeses and vinegar and bread and beer and a variety of fermented materials. We even produce enzymes needed in manufacturing in the pharmaceutical industry. We can make cellulase that is used in many applications, and also insulin from a bacteria. We now make insulin from bacteria. We used to have to get many piles of pig pancreases to make just a little bit of insulin to give to people. All right. Microbes can have a lot of impact on us. They are, microbes do produce things for treatment like insulin. It's very important that we know about microbes. They can prevent food spoilage, knowing that we need to keep our food at a certain temperature. If food gets too hot, if it sits out at room temp for too long, the bacteria will grow in it. They will release toxins, and this can make us very sick, lead to spoilage, causing disease, and all kinds of things. We got aseptic techniques. If you watch some of the videos that I did on diseases, we talk about puerperal fever and how people got it because doctors didn't clean themselves after dissecting cadavers and then would deliver babies. Contamination in medicine and laboratory technologies, laboratory medical lab technologies, microlaboratories, microbiology labs, things like that, medical labs, things. We can prevent contamination so that we do things. All right, so let's talk about some outbreaks. So a big outbreak, influenza. That's a big one. Okay, influenza called by influenza viruses. So viruses, there are two common ones: influenza A and influenza B. If a person has this, you're going to see some fever, coughs, sore throats, headaches, lethargy, fatigue, body aches, runny nose, things like that. Some respiratory issues, things like that. 1 to 2 weeks, it can be very mild, it can be severe, but there are vaccines against influenza viruses. A big important thing, the 1918 Spanish flu killed a lot of people right after the end of the First World War. The Spanish flu epidemic was really big. If you go to that time frame and you go to a particular cemeteries, and they have graves of that time, you will see loads of graves that say the infant son of, the infant daughter of. Really wiped out young ones, young and old especially. killed a lot of people as a virus outbreak. We hadn't seen anything like that for 100 years till we saw COVID. COVID was kind of like a 100-year pandemic. Ebola. Ebola always going over in Congo. Congo has a lot of problems with Ebola. I don't know if there is still another. The West African outbreak was the worst we've ever seen of it in Guinea, Liberia, Sierra Leone, Nigeria. It's a viral hemorrhagic fever, pretty nasty, internal bleeding, even some external bleeding and clotting. You bleed out your holes. You come in contact with body fluids. As of April 2016, when some of this stuff, like the book was written that I originally wrote some of the notes from that I first started writing when I first started making micro, and I want to update some of the stuff. There was a lot of cases. There's about 28,616 cases with 11,310 deaths. And that was in 2016. It has somewhere between a 50% to 90% mortality rate, so pretty high. There's no vaccine for Ebola. This is the classic shepherd's crook Ebola virus image. One of the things about viruses is this, guys. Many of them have vaccines. It's very hard. This is an RNA-based virus, so hard to deal with. Measles, extremely contagious is measles. Measles is basically this respiratory disease that gives skin rashes. So symptoms include a cough, runny nose, but a rash. It starts at your hairline and goes down your body. But we have the MMR vaccine. Now, you at this point, the MMR vaccine, I got, of course, 2000 we eradicated it with it. But unvaccinated individuals are causing it to spread. It's spreading right now in America. Now, this vaccine does not cause autism. There's absolutely no mechanism that shows how this vaccine causes autism. It does not cause autism. The rise in autism is changes to our ability to detect. If anything, it has to do that we are better at finding it. Okay, we used to have autism. Later on, we're going to talk about Anton von Leeuwenhoek, who made over 400 microscopes. If that guy basically wasn't neurodivergent, I don't know who was. I don't know anything. If some of these scientists of the early days weren't basically autistic, I know nothing. Okay, so anyway, let's talk about some other things. Hepatitis is a big viral. There was some cases in Tennessee this last year. That's out of date. I need to fix it. I try to update some things. I wrote this stuff. Basically, kind of the story is I was going to be teaching micro, and then I wasn't. I was going to be teaching micro, then I wasn't. I had training in micro, and I could teach it, and then I wasn't. And then now I'm teaching the online micro. And I would write stuff and things like that, and put stuff from, make it real to them. So it's a viral liver infection. It's hepatitis, hepatitis A. You eat food, water. So basically your food, water, fecal contaminations, things like that. Mild, severe cases. There were 13 deaths in Tennessee when I wrote this PowerPoint. 2,250 cases of it. There were cases linked to McDonald's in Johnson City and McAllister's Deli in Kingsport, Tennessee, where I'm currently in Blountville, Tennessee, where I'm recording. I live in Johnson City, Tennessee, and frequent Kingsport. There is a vaccine available. If you get it with 2 weeks of being exposed to it, it's effective. But you can get the hepatitis vaccination, hepatitis A. Not all hepatitis has vaccinations. One that is weird is called red tide. Red tide is an algal bloom, so this is algae. Now this algae, when the temperature goes up and there's increased nutrients in the ocean, it comes up, Carenia brevis. The ocean life dies. What happens is this produces a neurotoxin. It infects shellfish. We collect the shellfish, we eat it, and we get sick of it. So this actually causes respiratory irritations as the ocean churns. There was declaring emergencies in places that have this red tide, tourism. The places that have red tide are oftentimes beach areas where people go there to go to the beach. Salmonella, the bacterial infections. Definitely can be found on lizards, turtles, things like that. Food products, salmonella. I, as a kid, thought basically it was just mayonnaise. There was a bioattack in Oregon in 1984 by salmonella being spread at salad bars by a cult. Nobody died from it. There were many hospitalized. There were 751 cases. A cult in Oregon actually was spreading salmonella to salad bars in the area. Major outbreaks of E. coli 0157H7. That is a hard set of numbers for me to remember. In 2019-2020, you may remember the romaine lettuce outbreak during right before COVID hit. So, all right. So there's some emerging diseases we might talk about there. Now, these diseases will be on the test. Let's talk a little bit about nomenclature. There was a guy named Carolus Linnaeus who basically was like, let's organize things. One of the things that scientists love to do is classify and organize. It's called nomenclature. We classify. Now, we classify things, and it's something that humans like to do. We look at the way somebody is dressed, the way they act, the people they hang out with, and we say, you are a jock, you are a nerd, you are an emo, based on how you're dressed, how you behave, and who you hang out with. Well, we do the same thing with animals, with organisms. We give them a name, and it's two names. We call this a binomial nomenclature is actually what we call it. Is a genus name and a specific epithet. We will talk about this a little bit more later, but right now I'm going to go ahead and talk about the genus and specific epithet, the species name. We always italicize it and underline it. The genus is capitalized, the first letter. The specific epithet, and everything else is lowercase. Okay, you capitalize the first letter of the genus name. For example, Escherichia coli. Capital E, all the rest of it lowercase. It's Latinized. It's standard. And it's usually, it might be descriptive of what it looks like. We might use luteum or albicans for white or yellow, the color it turns, color it has. We might go where it's from, carolinensis, something about it. We might name it for the scientists who discover it, like Theodore Escherick, okay, Escherichia coli, okay. So we might describe the species habitat, Escherichia coli, Theodore Escherick, and coli, the colon. Scientific names can get abbreviated. So Escherichia coli, we would say E. coli. After we use it once, we go Escherichia coli. Then we would come back and say E. coli. Staphylococcus aureus, S. aureus. Okay. All right. Now, there's many kinds of microorganisms. There's bacteria, which is going to be a big part for this class. Archaea, we talk about them. They don't cause diseases. Fungi, protozoa, they definitely cause diseases. Algae, some of them like algae, the algal blooms that cause red tide, that can cause neurotoxins and poisoning. Viruses, definitely. Multicellular animals like parasites, like ticks, mosquitoes, biting flies, things like that. We will talk about them because they're vectors of a lot of diseases. Biting sand fleas, lice can transmit typhus, things like that. Worms can be infectious, things like that. So there may be infectious or vectors. So things like that. Okay. So a big thing that always is on my test, if it ever comes out of my mouth, it will be on a test, is the difference between prokaryotes and eukaryotes. Always on my test. For a microorganism can be a prokaryote or a eukaryote. That's it. Prokaryotes, pro means before, karyon means nucleus or pre-nucleus. These are cells that don't have a membrane-bound nucleus. They don't have any organelles. They're not going to have a Golgi, or they're not going to have an endoplasmic reticulum. They're not going to have that kind of stuff. These are bacteria and archaea. Eukaryotes, these mean U means true and karyon means nucleus, so they have a true nucleus. They're big cells. They have a membrane-bound nucleus, and they have organelles. So they have things like Golgi apparatus, endoplasmic reticulum, all that good stuff. Okay. Now these guys might be fungi. They might be protozoans, algae, multicellular animal parasites, your worms, your parasitic biting flies, ticks, things like that. Your parasitic worms and multicellular animal parasites. Okay. Now bacteria, these are prokaryotes. One of the big things about bacteria is they have a carbohydrate in their cell wall called peptidoglycan. Pay close attention to that. You will see it multiple times throughout the semester, is peptidoglycan. That is a target for some antibiotics and things like that. There's a peptidoglycan in the cell walls for things like penicillin and its relatives. They reproduce using what's called binary fission. One becomes two. For energy, they use organic chemicals. They use inorganic chemicals. They may use photosynthesis to do this. They take on many shapes. For example, rods, spheres, and corkscrew. Bacillus rod, coccus spherical, spiral corkscrew. They may or may not have flagellas. If they have a flagella, they have it for motility. We have archaeas. Archaea, they don't have peptidoglycan in their cell walls. A big difference is they have pseudomurin. We sometimes call that pseudopeptidoglycan. They live in extreme environments, so we call them extremophiles. They can include the methogens that create methane, the halophiles that love salt, and the thermophiles that love heat. They are not known to cause diseases in humans. Okay. Eukaryotes can be things like fungi. Fungi are eukaryotes. They have a carbohydrate in their cell walls. This is called chitin. Chitin is what we find in arthropods' shells. In the exoskeleton of an arthropod, we find chitin. Chitin is a polysaccharide. Okay. They use organic chemicals for their energy. These may be molds and mushrooms, multicellular molds, multicellular mushrooms. They have a mycelia, which is a mass of fungal hyphae. They can be unicellular like yeasts. They can reproduce sexually or asexually. They can have sex or they can bud and things like that. Protozoans, they are eukaryotes. They're unicellular eukaryotes. They use amoeboid movement. So they take these little pseudopods right here and they ooze them out and they pull. Okay, they move with pseudopods. Pseudopodia. We call this amoeboid movement. They absorb or they ingest their food, organic chemicals. They may have some pseudopods. They may have cilia. They may have flagella. They can be free living. They can be parasitic. They can produce sexually or asexually. So they do a lot of things. Algae, this algae here is Volvox. Algae are eukaryotic. They use cellulose in their cell walls, unlike fungi. So the only eukaryotes that have cell walls are fungi and plant algae. Okay, plants and algae have cell walls. Fungi have cell walls. Animal cells don't have cell walls. These can be everything from unicellular things like your diatoms to multicellular seaweeds and kelp. They do photosynthesis. They produce oxygen and other compounds, other organic compounds besides oxygen. They make sugars and other things. They live in fresh water, salt water, and even the soil. Some algae live in the dirt. Okay. I used to study an algae that lives symbiotically in salamander eggs. Viruses, they are acellular. They don't have cells. They actually are called non-living. The only reason we even count them, they evolved from living things, and they have to have a host cell to live into. So basically you have a DNA or RNA core and then a protein capsule around it, a protein coat, that may or may not have a lipid envelope. If they have a lipid envelope, it's really good to wash your hands to kill these viruses, like with SARS-CoV-2. Viruses only are considered alive when they're in your cells, and they are obligate parasites. They're intracellular parasites. They are obligate intracellular parasites because they cannot at all make it without going into a host cell and replicating in the host cell. They require a living host. So they break that what is alive definition. They are not alive. HIV, the virus that causes AIDS, it's a virus that breaks that thing you guys probably learned about called the central dogma of molecular biology. The idea of DNA gets transcribed into messenger RNA, and messenger RNA gets translated into amino acids to make polypeptides. That idea because HIV has reverse transcriptase. It can infect animals, it can infect plants, even bacteria and archaea. The deadliest virus on planet Earth is bacteriophages. They wipe out a bunch of different cells. So they kill multiple many organisms. Here we have a heart that is infected with diphtheria, a helminth, heartworm, Diphtheria emetius. Eukaryotes, multicellular animals, they're parasitic flatworms like flukes, tapeworms and flukes, and roundworms. Roundworms we call nematodes. These, so you have parasitic flatworms, parasitic roundworms. They're called helminths. Parasitic worms, flatworms, and roundworms. And they have microscopic stages like microscopic eggs, microscopic larvae. They can affect the heart. This is Diphtheria emetius. Today I pledge to do my part for optimal marriage fitness to reside in one another's hearts like Diphtheria emetius. You probably are familiar with the caduceus, the medical symbol for the medical professions. They believe it's actually a procedure for removing guinea worms out of the skin. Is out of the sore of the guinea worm, you would wrap it around a stick, and that's actually guinea worms. Doctor removes guinea worms from a patient by winding it onto a stick. We think that's where the caduceus came from, the medical symbol. Classification of microorganisms. We have domains. Do kings play chess on fiberglass stools? Domain, kingdom, phylum, class, order, family, genus, species. There's domains. Bacteria, archaea, eukarya. Eukarya has the protist, that's called protozoa and algae. The fungi, that's things like yeast, molds, mushrooms. Plants, those are like mosses, ferns, conifers, and all your flowering plants. Animals, that's things like everything from sponges to us. Worms, insects. Many animals are vectors for disease. Plants can be hosts for disease. Fungi can cause disease. Protists can cause disease. Bacteria can cause disease. Now, what we're going to do is we are going to stop here. Normally, I wanted to stop at 32, but we have the history of microbiology. No, I'm going to stop. Never mind. I'm going to stop at that second history of microbiology. So, a little bit of history of microbiology that I want to talk about is this idea of cell theory. Two big names. Now, these people that did cool stuff will be on my test. The dates won't be, but the scientists will. The date might be in the description. If you're really good with dates, definitely use it, but I'm not. I won't have the dates. About 1665, this gives you guys quite a long time. There's a guy named Robert Hooke. Robert Hooke basically had a microscope, reported that living things were little boxes called cells. It looked like the cells that monks lived in. And then 1858, quite a bit later, Rudolf Virchow said, hey, cells come from other cells. And that gave rise to cell theory. Cell theory says four things. All living things are made of cells. Cells are the building blocks of life. It's another way to say it. All living things are made of cells. Or to say, cells are the building blocks of life. All cells come from other cells. Virchow. Cells are the smallest things of life. The smallest thing that actually has the properties of life is the cell. take General Biology 1 or A&P 1, and you will know about what makes life life. And each cell maintains homeostasis. That's the tenets of cell theory. Okay? All right. Now, so you have archaea, bacteria, eukarya. They have some differences, and there's a lookup about their differences and things like that. Some things I don't worry about, some things I do worry about. Okay? Now, we talked about the nomenclature, the naming system. Taxonomy is the science and discipline of doing basically what you did in high school. You looked at the way somebody was dressed. You looked at the people they were with. You looked at how they acted. And you were like, they're a nerd. They're a jock. They're an emo. We had goths. I don't know what the devil you guys had, but we had goths. Taxonomy, we would use classification, we would use characteristics that they have and say, you're this. Taxonomy. It provides names, those universal names, the binomial nomenclature I call it, for reference. Again, why do we do that is, for example, guys, if I'm talking to a scientist from China, and I'm talking about them about a species, for example, I was talking to somebody from China about a species of salamander, Cryptobranchus alleghaniensis. That's the hellbender. We call them hellbenders. Snot otters. They didn't know what a hellbender was. They didn't know what a snot otter was. But they know Cryptobranchus alleghaniensis because they read the literature. So we have to use that sometimes to communicate from people from other places. Phylogenetics. This has to do with evolutionary biology. It's grouping organisms based on common properties of common ancestry of evolutionary history. It's making, and a phylogeny is an evolutionary history. It implies that everything evolved from a common ancestor. We use ribosomal RNAs, we use anatomy or morphology, and we use fossil evidences to come up with evolutionary histories. We use molecular, structural, and fossil evidences to come up with those. Okay. All cells have ribosomes, so, alright. Do kings play chess on fiberglass stools? Domain, kingdom, phylum, class, order, family, genus, species. Guys, you go to the club, you see a good-looking girl, you go up to her and say, girl, your name must be phylum because you are above class. Okay? Now, what we do is like the sorting hat from Harry Potter. That's probably a little bit before your time, so I apologize. We're sorting people out. Domain is incredibly inclusive. Domain eukarya has everything that has a nucleus in its cells. Domain bacteria is everything that has peptidoglycan and doesn't have a nucleus. Domain archaea is everything that has pseudomurin and doesn't have a nucleus. Now, we have Saccharomyces cerevisiae, the baker's yeast. It's a fungus, eukarya, fungi. Ascomycet, which is Ascomycota, which is sac fungi. And then you go down, 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 down. Okay, until you get down to Saccharomyces cerevisiae, the species. The genus and the species. Saccharomyces cerevisiae is the Canis familiaris. That's your dog. Alright. Do kings play chess on fiberglass stools? Okay. Alright, prokaryotes. If we have prokaryotic species, we can culture them. We have a population of cells. We can culture them, cells grown in a laboratory media. We can clone them. We have a population of cells from a single cell. I get one cell and then I clone it, and that's a clone. Like, I got a clone of this. I pick and transfer and I keep my clone. I've cloned bacteria. Strain is a genetically different cell within a clone. So there's a strain. Strains can give us unique abilities. we can classify viruses. Now, viruses are thought to have come from something that was alive, evolved from something alive. Okay? Viral species, the population of virus, they may have similar characteristics. We might look at their ecological niche. What do they infect? How they infect? A variety of things that we use to classify a viral species. So we actually do give viral scientific names. It's just they evolved from something alive, and we can do evolutionary phylogenies on them. But viruses are freaky deaky, man. They're weird. Okay? All right. Now, kingdoms of eukaryotes. There's domain eukarya, and there's the kingdoms: Animalia, Plantae, and Fungi. Okay? And then Protista or Protists, they're kind of like, hey, I don't fit the other kingdoms. I'm a catch-all kingdom. If I don't fit in an animal, plant, or fungi, then I'm like an animal-like protist, a plant-like protist, or fungal-like protist. And you're usually a single-celled eukaryotic thing. Okay? Animalia is multicellular. There's no cell walls. They're chemoautotrophic, which means they eat food other than themselves. They have to go eat something else to eat, like eat plant, eat animal, eat meat, eat plant matter. Plantae are multicellular. They have cellulose in their cell walls, and they're photoautotrophic. They make their own food. We say usually because there are actually some non-photosynthetic plants, like bear corn, for example, is non-photosynthetic. Fungi are chemoheterotrophic, which means they eat other stuff. Chemoheterotrophic means I get my food from something else. They can be unicellular like yeasts, multicellular like mushrooms. They have chitin in their cell walls. They have spores or some kind of hyphal fragment. Protists, that's the catch-all. Eukaryotic organisms don't fit in the other. It's based on clades. Clades are groups that contain all the living and non-living things. The common ancestor and all the living ones. Alright, we're going to stop here, and we're going to start off with next video with the history of microbiology and some other diseases. Alright, see you in the next one.